Rare Disease Day is tomorrow, and it gives us a chance each year to focus attention on the millions of Americans and the support givers, their families, who are living with rare diseases. I'm pleased that so many rare disease advocates have come to Washington to make their voices heard and rally support around these issues. We look forward to continuing to work with all of you to keep up this important work. It's good work, but important work. And as we continue to work here in Washington, and that is what we still do, I'm told. Leonard Lance has joined us as well, the co-chair of our caucus, so from the Republican caucus. Thank you, Leonard, for all your help. As we continue to work here, that we don't lose sight of what the important issues are, and amongst them is finding cures and discoveries of cures for people who are suffering with rare diseases, oftentimes the orphans, in terms of research and discovery. We need to put as much focus and attention in a realistic way on these issues as we do on the much bigger issues as well that our country is facing. And that goes from NIH to tax policy as well. More on that in the days and weeks to come. But that really, once again, thank you for holding this forum, and thank you for having me here today. I look forward to meeting a number of you throughout the day today and tomorrow as well. So God bless you. Keep up this great work.
Congressman Lance, it's a pleasure to welcome you to the podium.
Thank you very much. Thank you. Good afternoon to you all, and thank you for being here in Washington, up Pennsylvania Avenue on Capitol Hill. It's rather like Baker Street in London, and I'm certainly honored to be the Republican chair of the caucus, and Congressman Crowley does an excellent job as the Democratic chair. It just shows you that we work together, Republicans and Democrats. We work together, New Jersey and New York. Can you imagine that? And we certainly work across the country. This week we recognize the work of the tireless advocates for rare diseases, many of whom are in this room. And I have the honor of serving as the Republican chair of the Congressional Rare Disease Caucus, as Joe is the Democratic chair. And I consider our work an important responsibility as part of our service here, and it is an honor to work for innovative treatments, new technologies, and to build an atmosphere of appreciation and understanding on Capitol Hill for the hard work of all of you, the patient advocates. Your passion is often driven by a care for loved ones, and your personal stories are profiles in courage. Hearing from countless advocates, many of whom are here this week in Washington, gives the members of the caucus renewed energy and purpose. Events held during Rare Disease Week highlight what has been accomplished and what still needs to be accomplished. And there's a lot more to do, but we'll do it together. I am working on important legislation in this area, the bipartisan Modern Cures Act, and thank you for your unanimous support of that. And as you know, it would update the nation's drug evaluation process to encourage the discovery and development of new treatments for chronic and rare diseases. The measure will also create a system that rewards efficiency and effectiveness to the benefit of all persons with rare diseases. The Modern Cures Act will encourage the development of drugs abandoned in the development process. It will create a new category of drugs known as dormant therapies for compounds with insufficient patent protection. Drugs that offer the promise to treat conditions with unmet medical needs. Updating regulatory networks such as patent reform will help open the pipeline for new innovations and therapies. Patients with degenerative conditions, cancers, and rare diseases await the genius of these new solutions. While we do not know the cause or cure for many of these rare diseases or cancers, we do know that awareness is the best protection, information is the best tool for innovation, and well-rounded care during and after treatment is the best therapy. This is our mission in the bipartisan Rare Disease Caucus. to work together to find solutions that make a lasting difference. I again thank you, the families and the advocates, whose challenges we in Congress may never know completely, but whose commitment to your loved ones is unyielding and inspiring. The caucus pursues its mission in your name. Welcome to Washington this week. Welcome, of course, tomorrow in the day that is set aside, the day across the year that is set aside in this noble cause. You've brought cold but good weather. I understand there's a snowstorm coming Sunday. May you safely return across the nation as together we continue our unyielding effort to make sure that we will continue the progress that is essential for the health of the nation and, might I suggest, for the health of the world. Thank you very much, ladies and gentlemen.
It's my pleasure to be here today to moderate. Now, some of these speakers are not moderatable, so I just want to put that out there at the beginning. But it's so interesting to me. Once upon a time when I worked at NIH, we used to talk about the continuum from basic research through translating the research results into reality for patients to the bedside of the patient, where the healthcare provider and the family realize the benefits of all of that work together, and the patient, most of all, realizes that work. And the way that this briefing is laid out follows exactly along that idea, that we start with these ideas that are nurtured by research and by a better understanding of science and a better understanding of the disease and how the disease happens to people and changes people. And we then build on those ideas, principally in the pharmaceutical industry, to try to develop those ideas into a product that can treat those patients about whom we learned from that basic research. And the end for all of us is better health care and a better chance and hope for a better future. I think in the area of rare diseases, we more than anyone understand the reality that that pathway is not yet a perfect one, because we have so many conditions where there is so little understanding and really very small hope, but there is never zero hope. And today we are going to talk about how we are thinking about making that hope a reality for every family and every person affected by a rare disease. So we're going to start at the NIH end of this spectrum with Dr. P.J. Brooks, who is in the Office of Rare Diseases at NIH, and that office is part of the new National Center for Advancing Translational Sciences. And this, I don't know how much all of you know about NIH, but the notion that NIH, the bastion of basic biomedical research, now has a Center for Advancing Translational Sciences is a huge breakthrough in and of itself. And Dr. Brooks is going to make that a reality for all of us in this room. Thank you.
Well, thank you, Kay, I think, for that introduction. So yes, I do work in the Office of Rare Diseases Research at NCATS, and it's a very exciting place to be at the NIH, focusing on translational sciences as someone working on rare diseases. And so what I'm going to do today is what the caucus asked me to do, is work with Marshall Sumner, who's an expert in rare diseases, and kind of give you a little bit of Biology 101 as kind of to sort of set the groundwork for the discussion we're going to have. I've got some slides here they've asked me to cut down, so I'm going to have to zip through them fairly quickly. I'm also going to have to look back behind them to see them, but I'll do the best I can. So if I can have the first slide. Okay, so a lot of what we'll be talking about here are genetic diseases, and we talk about the human genome. Basically what we mean by that, or the way to think of it is, it's sort of the instruction manual for making a human being. And those instructions are encoded in the DNA, the genomic DNA that in humans is separated into 23 chromosomes. We each get one set of those chromosomes from the mother and one from the father, so we got 46 chromosomes that exist in the cell within the cell nucleus, as indicated on the slide. In addition to the cell nucleus, there's a small bit of DNA, a small genome in the mitochondrion, which is the energy factory of the cell, and so it has its own genome, and so together the nuclear and the mitochondrial DNA are what we think of as the human genome. Next slide. So the DNA, as you probably know, is classically been identified in the structure as being a double helix. It's identified by Watson and Crick in the 1950s. And the structure is fascinating, but what's actually more important from the standpoint of information transfer is what keeps the double helix together, and that is base pairing. And so the base pairing is shown on the slide here. The rules are such that there's basically four bases, four letters in the DNA code. We call them A, T, C, and G. They stand for the four nucleotides in DNA. And the way the base pairing works is the A base is always paired with the T, and the G base is always paired with the C. And that's really important because what it means is that if you ever have one sequence or one strand of the DNA, you always know what the other strand is by using the base pairing rules. And then the other kind of nucleic acid we'll be talking about, which is single-stranded, is called RNA, and that acts as a messenger and does other processes within the cell. Next slide, please. So when we think about a gene, essentially the way to think about it is a gene is a piece of the DNA, a piece of genomic DNA that contains the instructions to make a protein. And we have roughly 25,000 or so genes in the human genome. And so the process of going from the DNA to a protein is indicated on the slide. It involves basically two steps. The first step is going from the DNA to another molecule, the messenger RNA. And that process is carried out. That process is called transcription and takes place within the cell nucleus. That messenger RNA then leaves the cell nucleus and goes out into the cytoplasm where it encodes the proteins. And the proteins are basically strings of amino acids attached together. And proteins carry out various functions in the cell, and I think Marshall will talk more about those. They may be enzymes, they may be structural proteins, they may be hormones, for example, like insulin. So basically they carry out the functions. And that process is we refer to it as gene expression, and that DNA to RNA to protein is also often referred to as the central dogma of molecular biology. Next slide. So I think I'm going to skip this one. And skip that one. All right, so well, I didn't anticipate doing it this way. So when we talk about genetic diseases result from mutations in the DNA sequence. And so what you're seeing on this slide is first up above is a normal sequence of a gene encoding a protein. And you can see that the different amino acids in the protein are encoded in sets of 3 triplets. We call those triplets codons, and this is the triplet code. So that particular protein has a certain series of amino acids, and that's the normal series. But what I've shown you below is some different types of mutations that can occur. So in the second one, instead of the serine as indicated at the TCA, any chance you have a pointer? Okay. Oh, good. Yeah, you know this. Okay. Instead, there's a C base got changed to an A, and that causes the protein to stop prematurely. So you get a small piece of protein that doesn't really carry out any function. The next type of mutation down below is what we call a missense, where you've made the wrong protein. You've got a proline for a serine. And then finally, the one all the way down below is called a frameshift mutation, which is sort of the worst case. You've removed one single base, and now the whole reading frame is changed off. When the protein goes to read in those sets of 3, everything is off by 1, and you make a totally nonsensical protein. And I'm kind of pointing on this because different types of mutations have different implications for therapeutics. There are certain drugs that we can test, for example, if you have a stop codon mutation, as one example. Next slide. So one of the things that is worth keeping in mind as we think about disease and therapeutics is the process of going from the DNA and transcription into the protein. When I first showed it to you, it was sort of a linear stretch of DNA directly encoded the RNA. But in fact, in mammalian genes, it's a little bit more complicated than that. The protein coding regions are actually broken up into stretches that we call exons, as indicated by the slide, and those are separated by stretches called introns. So when the messenger RNA, when this RNA is first transcribed, the introns have to be removed and the exons have to be spliced together. And that process actually, and that process has to be done very precisely so that that reading frame is maintained and that there's no chance of making a mutation that would make a non-functional protein. And this splicing process actually allows the same region of the gene to encode potentially more proteins because you can sort of mix and match exons and make multiple types of proteins. Next slide. But also there are mutations that can affect the splicing process. And so introns that might normally have been removed are left in, or exons that should be left in are taken out, and that again makes non-functional proteins. And here again, for splicing mutations, depending on the exact nature, there are certain therapeutic possibilities that arise depending on exactly what kind of mutation it is. And particularly for splicing mutations, in some case we can go in and modify the splicing process in a therapeutic way. Next slide. Oh, and that's just an example that splicing mutations affect literally hundreds. You can see splicing mutations in hundreds of rare genetic diseases. Next slide. All right, so when we talk about genetic diseases, there's basically 2 different types, 2 fundamentally different types. There are recessive diseases and there are dominant diseases. So in the recessive disease, as indicated here, each parent contains or is carrying a normal gene and a mutant gene. And so their offspring, there's about a 25% chance that one of the offspring will inherit both mutant copies and be affected with a disease. And the idea of it being a recessive disease, the kind of key aspect there is that it's a loss of function. Some protein that should have been made in the cell is now no longer made, so something is missing. And that's what we call a loss of function disease. And many of the genetic diseases you're familiar with are recessive cystic fibrosis, Tay-Sachs, many of the most common ones. When I study xeroderma pigmentosum, which is a DNA repair disorder, many different recessive diseases. Next slide. So the other type, the other fundamentally different type, is called a dominant disease. And in this case, one of the parents is carrying the disease mutation. And basically, in this case, one mutant gene can cause a disease. And so in that case, about half of the children will be affected. And the dominant disease, you can basically think of it as a gain of function. So the protein is made, but it has a toxic function that causes damage to the cell and causes cells to die. And one of the most well-known examples of a dominant disease is Huntington's disease that you may have heard of. Next slide. And these 2 diseases we like to think of them as in this way because when we're looking at rare genetic diseases, rare diseases in general, we have thousands of them. And thinking about trying to treat thousands of diseases, each one individually, is rather daunting. We certainly try, and in some cases we succeed, but it's a rather daunting task. So what we're always interested in is how can we group diseases in some way that will allow us to develop a single sort of therapeutic modality, a platform, if you will, that could be used to treat multiple diseases. And so thinking about this dominant and recessive difference is sort of important, because if it's a recessive disease, you're missing something. And so one strategy is to put that missing something back in. So if there's you can put, for example, a normal copy of a gene back into the right cell through gene therapy, or you can put an enzyme back in the cell by enzyme replacement therapy. And those are both strategies that are generally applicable if we can get nucleic acids into cells or proteins into cells, for example. With the dominant disease, it's sort of the opposite. You want to knock down or somehow inactivate the bad gene, and we have ways to do that too, nucleic acids that will degrade the bad messenger RNA. So we're very interested in these platform therapies. Okay, and then finally to sort of wrap up here, the question comes up, we've got a single genome, we've got multiple different cell types. How does that genome make all those different cell types? And the short answer is genes can be either on or they can be off. And so in a brain cell, for example, the brain cell genes are on, the pancreas and kidney genes are off, and that's what makes a brain cell. So we've learned a lot about, next slide, how genes can be turned on and turned off, and the process, or the general term we use for that, is epigenetic regulation. And one way you can shut genes off is there's an enzyme that puts a methyl group onto the cytosine, as indicated there, and when the cell recognizes that methyl group, it brings a set of other enzymes, some of them called H-stacks, as indicated behind me. And those H-stacks act on the chromatin and other proteins surrounding the DNA to shut it down and turn off gene transcription. And that's significant because sometimes those processes can turn off genes that we wish were on and cause disease. And so there's a lot of interest in epigenetic diseases and drugs to address epigenetic disease. Next slide, and I think I'm going to basically just stop here. Thank you very much for your attention. I'll be happy to participate in the discussion.
Okay, raise your hands if you understood that whole thing. Oh, look at that! That's good, good. And then when we all go to have our genome sequenced, we're going to just know exactly what it all means because we have the picture in our mind. Our next speaker is Dr. Marshall Summer, who is the Division Chief Genetics and Metabolism at Children's National Medical Center. Dr. Summer is a specialist in rare diseases, and the only thing I want to say about him is if you're lucky enough to work at Children's National Medical Center, you're lucky enough. Dr. Summer.
So why am I giving this talk? First, I'm from Tennessee and I speak slowly, although that won't be an option today. I've got about 30 years of experience working with patients with rare diseases and also doing bench research as well. I work with the National Organization of Rare Disease, the NIH, the FDA, Children's National, and I'm a cheap date because I live in the district, so I don't cost anybody a plane ticket. When most folks think about rare disease, this is what they think of, sort of some obscure mad scientist living in a laboratory doing things that no one really wants to hear about. That actually includes the geneticists themselves. We kind of like that sort of mystique about ourselves. But the other thing to remember is when you start talking about rare diseases, what's the largest category in almost any survey you do? It's other. And really that's kind of how rare diseases have slotted in. They are these 7,300 plus rare conditions that there aren't any, large numbers in any one group because they're rare, but also when you take them as a group, it's a huge number. This is also what most people think about. Yeah, we probably should have included coffee with lunch. But when you think about all the different things that can go wrong biochemically, genetically, all of the different impacts this can have, this is kind of what a lot of folks think. This is also why a lot of folks try to avoid this field. This, however, is the Tokyo subway system, which looks very much like the biochemical wall chart I just showed you, but the thing to remember is 15 million people navigate this every single day. And even though these rare diseases are complex, they have complex interactions, you can actually find your way around these with a good map. All right, this is a concept I'd like all of you to keep in mind. We call this environmentally determined genetic expression because we wanted to use the word edge when we were describing it. But the basic idea here is the rare diseases we have are way up there in genetic severity. So when they have the clinical expression, the clinical things that go on with them, it's usually because there's a very severe genetic change, which we just talked about. And without very much environmental interaction, folks actually will have the disease. Now, if I go out and stand in the middle of the beltway here, well, actually not at rush hour because the cars move slow, but when the cars are moving fast on the beltway and I get hit by a cement mixer, it doesn't really matter what my genetics are. I'm going to have a problem no matter what. But most folks live in the middle. They have mild genetic changes. They have mild environmental exposures. One of the things we've learned from the rare disease field helps every single us. Our rare disease patients are really some of our best teachers about medicine, because what we see in these extreme genetic situations actually teaches us about more common things. I'll give you an example of that in just a minute. So no matter where you are in the field of medicine and biology, rare disease is highly relevant. It really is one of our best teachers about health, about human physiology, about the use of genetics, therapeutics, you name it. So one of the reasons I was drawn to this field was for that very reason. We're actually able to help out the rest of our physician colleagues dealing with more common things by working with rare disease. And just cycle on through that one to the next slide. All right, so keep going. So the rare disease field has kind of become quite popular. When I first started off in this, it was kind of like, yeah, you're the other guys, you're the ones out there. But what we're finding now, it's becoming one of the hottest therapeutic fields. Why? There's probably 25 to 30 million Americans with rare diseases. There are really over 7,300 currently cataloged at the NIH. And it hits folks of all ages, all types. Historically, we thought of them as pediatric diseases, and that's still kind of the most common mindset we have about them. But my oldest patient we diagnosed with a rare disease was 94 years old, and that was her first presentation. So we see things at all ages and all group size. So the stadium's kind of gotten filled up with a lot of the things Congress has done and the FDA has done. It's become a more popular field to develop therapeutics in. So we're actually getting more interest than we have at any time, at least during my career. Now, here's an interesting fact. Rare diseases may be rare, but as an impact on child health, they're quite large. If you go through any pediatric ward at any children's hospital in the USA, 25% of the patients will have a genetic condition. Those 25% of patients, however, represent 43% of the hospital costs and billings. So as a group, the impact on healthcare is much higher than patients with asthma or patients with a common infection, things like that. Our patients have serious diseases. They end up using a lot of healthcare resources. Some things to remember, if you're going to play in the rare disease field, the family groups tend to be very, very active. We tend to have groups much more active than you would in, say, something like hypertension, heart disease, things like that. They're actually a lot of fun to work with. One of the moms compared herself to a wolverine. I had to go look that up because I'm from the South and I didn't know what that was. The other thing to remember is when you're working with patient groups, there aren't many. So you actually have to use different models for study design. You can't do a hypertension study with 1 million people in it because there don't exist 1 million people with these conditions. The classic definition is under 200,000. Most are well below that. I have a daughter with Down syndrome, and as my wife puts it, the other doctor summer, she said that's almost rare. But there are not many patients, so you have to use different models, different standards for how you determine efficacy of treatments, things like that. The other thing is you're going to be dealing with a lot of academicians when you deal with this field. We tend to be a little goofy. Sometimes we wander mentally astray. And by the way, you did a nice job on that update on molecular biology. I may steal the slides from you later on. But this is the kind of stuff we talk about all the time. We live, eat, and breathe this stuff. And unfortunately, it means sometimes our colleagues aren't quite as tuned in with what we're doing there. Things that are really helpful, the NIH, FDA, and Congress has done a lot to help us out in the field. The NCATS program has been incredibly helpful, mainly because some of the biggest therapeutic advances in the last 20 years have been in rare diseases. In the field I work with in urea cycle disorders, with fewer than 1,000 patients, we've been able, working with NIH, FDA, to bring 3 drugs all the way through the FDA process in the last 10 years. I think that's got to be some kind of record for doing that sort of thing. But also, we're gaining better understandings on our patients. One of the things to do when you're trying to figure out how to treat a patient, when you're trying to figure out how to improve their life, you have to actually understand what's going on with them. And a lot of times if you only have a handful of patients, it's hard to know. It's hard to know how often do these patients get infections, how often do they have some of the complications they have. So one of the things we've done working with NIH and other groups like NORD is working on registry projects. By registries, what I mean are long-term databases where we're finding out what happens to our patients. So, for instance, if you develop a new treatment and you want to see if it's effective, if you don't know how common the thing it is you're trying to treat, you can't really figure out if the drug works or not. So one of the key steps we're working on and trying to pull all of these groups together is finding ways to do these long-term registries. Now, 7,000 different diseases, that's a lot of registries. So one thing we're doing a little differently from our classic model is we're involving the families and the patient organizations to help us do these registries. It's the only way economically we can do them. I think it's probably one of the best things we've got going. The most rewarding thing from my standpoint, not working for the government, is we've got the FDA working with us, we've got the NIH working with us, we have the parent organizations, National Organization of Rare Disease, all sitting down at the table trying to do this. There are very few fields in science or medicine where you see this level of cooperation and this level of support. And you guys are the reason we're doing this. So I'm going to actually keep us on schedule, start a little bit early, and then I look forward to any questions you may have later on, and thank you all very much.
To try to tie together what our first two speakers have talked about and point us in the direction of what's next after an understanding of this basic science, basic biology, basic understanding of the disease itself, is Dr. Emil Caucus. Dr. Caucus is the founding president of the Every Life Foundation for Rare Diseases, and he is also the CEO of Ultragenyx Pharmaceutical at the moment. But he has a very long history in working on the development of treatments for rare diseases. One of his first projects being the development of the drug Aldurazyme, and that was developed by him and then subsequently supported by BioMarin and Genzyme. Dr. Caucus is a legend in the rare disease world in his own right, and so I'm going to stop talking about him and let him talk for himself. Dr. Caucus.
Well, thank you all for coming today, and I'll talk a little about what we're doing at the Every Life Foundation. I've spent 20 years in the field developing rare disease therapeutics and started the foundation because I realized there are things we could do that would help the process between when a scientist has a discovery to treat a disease and when it can actually get to the patient. And that process does work, and the Orphan Drug Act, for example, has enabled several hundred therapies to get developed, but there's still 95% of the disease out there that don't have therapies, and there's definitely things that can get improved along that process. So we've been working on that, developing scientifically sound ways to improve the process of translating discoveries into science. And our view is that when a family is hit by what we think of as genetic lightning, when this event comes and you find out you've got something and your whole life and future is gone, whatever science we have somehow should be available for you to do something for your child and make that difference going forward. And unfortunately right now there's still science that's out there that hasn't translated, that is sitting there. It's been found and hasn't become a therapy, and we think that should be fixed, and there should be more work on translating these ideas and science into therapeutics for rare disease therapy, and that's what we're working on at the foundation. Now we work on that in many ways. We've been conducting workshops, working on legislation. We do work collaboratively with the FDA, NIH, industry, and patient groups as well, trying to find those steps forward, the things that we can do. I think those events include the need for incentives and include the need for improvement in the process and how we go about drug development. Incentives like the Orphan Drug Act are essential. The tax benefit incentives, the protection allows a researcher to say to a company that might want to invest, to say, look, there may be no patents, but if you get it approved, you'll have at least some protection to make that investment worthwhile. Making that work is really important in order to get them to say, okay, we're going to invest what will be millions of dollars for a treatment that might only help a couple hundred. And with the MPS 1 program, there's probably only a couple hundred patients in the U.S., just a few hundred in the world on the therapy now. But for the first 5 years I was working on that, there was very little help to be able to make that translate. It took an incredible set of efforts on our behalf from a foundation, the Ryan Foundation, a patient group, father, a policeman from Carrollton, Texas, who started a foundation and raised the money to help us survive and help that project go forward. Now today there's an example of how many challenges there are families, including Hugh Hempel here and others, that are finding the need to develop their own, and actually Jill Wood as well, developing their own drug development programs and forming corporations and raising finances. And the truth is, it means that we have a lot of work to do, because I think if the system isn't allowing the science to go forward and get these therapies developed, if we've done the billions of research at NIH to discover how to do things, and yet we're not giving it to people, and families have to develop biotech companies to develop their own therapies, and something's not quite right. Someone should be doing it for them, and something should be happening. Now, as a company I founded, Ultragenyx, we're working on 5 different diseases right now, and we're hoping to help innovate the strategies around development and continue to work at the foundation as well with Julia and the team there to push the envelope forward on how to make this process more efficient and how to incent development, so we make this translation of science into medicine as more effectively than we are today. So that's all I have to say from the EveryLife Foundation. I want to thank Julia and the team at EveryLife, who's really puts all this together, and the RDLA group that she runs for making this all happen today. So thank you.
I want to turn now to what I think the science is wonderful and necessary, and the translation of it is wonderful and necessary. But what makes the arena of rare disease work so incredibly rewarding, so driving toward commitment, is the families who work with this every day of their lives. And it's a great pleasure to introduce Hugh Hempel, who is the founder of the Addie and Cassie Fund, which he founded, named after and in honor of his children, his now 9-year-old twins, who have a rare disease. And as Emil already pointed out, a lot of the work toward getting somewhere to treat that disease has been done by Hugh himself. And I am so pleased to be able and so honored to be able to introduce him to you today.
First and foremost, the work that we've managed to do has been really done by a community of people, and I'm honored to be part of that community and here speaking on behalf of those folks. Most notably my wife, who literally works 17 hours a day on the subject matter. We started out as a research foundation. We were lucky, not lucky to be diagnosed with a horrible disease for our children, Addie and Cassie, who are now 10 years old as of January. And their life expectancy is probably in that neighborhood. So we've actually been successful in extending, I believe, their life. And at the moment, they're stable and relatively happy little girls. They're not verbal, they don't walk, but they're aware of their surroundings, and they know they're loved, and they give us a lot of love back. The work we do is, as I said, was fortunate. We came at a time when the basic research had already been done, and a combination of the NIH and through foundation money raised by amazing folks at Ara Parsegan Foundation, the DART, and families who raised the money to do the basic research. We entered the system in a place in time where a compound had been identified, a compound called cyclodextrin, and we grabbed that opportunity and went to the FDA and applied for an individual, essentially compassionate use IND to give our children this compound intravenously initially. We discovered that it wasn't working intravenously for the brain, which is where this disease my children have, which is like a childhood Alzheimer's disease. It wasn't reaching the brain, so we went back to the FDA and asked them to allow us to give this compound to our children directly to the brain, initially through a lumbar puncture in their spine in the back, which they get every other week now, have had 75 lumbar punctures to date over the last 2 years or so. And then subsequently after that, we decided that since the girls have to be anesthetized for the lumbar puncture, we want to install an Ommaya reservoir, which is a little port in the brain to allow us to dose them with cyclodextrin direct to the brain. And we installed those ports this past May, and almost a year ago, and I can report that despite a surgical complication for one of the twins, Cassie, Addie is now receiving treatment through her Omaya for the first time in the last couple of weeks. So it's been quite a journey. It's an odyssey. We were the first to do this for our children. I know that there are about a dozen other children now in the world, Japan, China, Brazil, who are receiving the same treatment using almost identical protocols to what we developed. So that's rewarding, to say the least. But almost more importantly, the NIH has undertaken through the NCATS program a phase 1 safety trial with 9 additional children here in the United States that's going along at the moment, with hopes to move to phase 2 once the safety trial is complete. So the good news is that the Orphan Drug Act, the work that Chris Austin and his team at NCATS and the NIH has undertaken, in combination with work that we've done in the community to raise money and do basic research, is moving a promising compound forward. It's not. I mean, it's just not enough. I mean, I'm proud to be here for the caucus, but I urge the caucus to continue to move the agenda forward. Modern Cures is a great step in the right direction, but even that can go further. And certainly we don't need to go backwards. The suggestion that we remove any tax credits that are already in place for this system is just mind-boggling to me. Those tax credits are a gnat in the overall tax credit system that really need to be there to incent the work we want to do. So I formed a small biotech, virtual biotech startup that I've been out trying to raise money from venture community, from philanthropic donors, and the only way that we can raise the money is if there is the orphan drug designation and the tax credits, because the overall economic picture is what makes this work. So I urge Congress to continue to press forward and do the good work that they've already been doing, and think larger and stay committed to the cause. I look forward to any questions and answers afterwards. Thank you for your time.
So that's going to be a hard act to follow. But there's one person who can follow it very well, and that's Jill Wood, who is the founder and CFO of Jonah's Just Begun Foundation to Cure San Filippo, in honor of her son, Jonah. Jill's foundation does research funding and awards grants, and Jill has worked tirelessly since her son was diagnosed with this condition to do exactly what Hugh was just talking about, which is move this forward toward a cure not only for her son Jonah, but for every child who is affected by a rare disease. So thank you so much, Jill, and it's a pleasure to bring you up here.
When Jonah was first diagnosed, it was the Hempels that I called up first to say, how am I going to do this? So I am walking in their footsteps right now. But it's hard to be introduced as a mom who started this company to save her son's life. I usually like to distance myself from it and say, I'm the founder of Jonah's Just Begun, a medical research foundation, and the CEO of a virtual biotech. But I'm actually here because my son is dying. Since PJ did all the science for me, I thought I was going to have to educate everybody on the science, but you took care of that for me. So Sanfilippo C occurs in 1 in 1.5 million births. It's caused by a genetic glitch. This glitch stops an enzyme from recycling a sugar molecule called heparan sulfate. Heparan sulfate is then stored in the lysosome of the cell. The storage of heparan sulfate causes catastrophic health problems. Our children appear typical at birth. In their preschool years, they start to show signs of the disease, presenting with behavioral issues, sinus and gastrointestinal issues, and speech delays, all of which can be disregarded as a phase a child is going to grow out of, which attributes to the late diagnosis of this disease. Next, the behavioral issues grow out of hand compulsions, hitting, aggressiveness, sleeplessness, hyperactivity. Cognitive delays are evident and can't be ignored. The odyssey for a clinical diagnosis begins. On average, it takes 2 years for clinicians to diagnose Sanfilippo. The disease is often misdiagnosed for years. So meanwhile, you're trying to figure out what's wrong with your child. The disease is attacking the central nervous system, causing profound brain damage. It doesn't stop there. The damage to the central nervous system causes horrific compulsions and behavioral issues. Children go weeks without sleep. They have no restraint, running into traffic, hitting and throwing, screeching on the top of their lungs. To some, they might appear mad. To your friends and family neighbors, they might assume that your child is spoiled or out of control. As the disease takes its toll, children lose the ability to walk and eat on their own, their speech long gone. Organs begin to fail. Many children succumb to respiratory issues like pneumonia. On average, death for Sanfilippo children occurs in the teens. Okay. I look out and I see. Stop. I'm not done. I see all these parents that are in my shoes, and I don't have to explain what this feels like. So put yourself in my shoes, my husband's shoes, and pretend you're us. Our new pediatrician at Jonah's first year well visit commented that Jonah's head circumference was off the charts and suggested that we take Jonah for an MRI. Our pediatrician recommended us to a pediatric neurologist, and the neurologist concurred with the findings and asked us to do an MRI. In due diligence, my husband and I took Jonah for an MRI. 6 months later, we actually had it done. A month later, the neurologist called us in to give us the results. At that point, Jonah was perfect. I mean, he was meeting all of his milestones. No idea what was about to happen to me. My husband just so happened to have the day off that day and joined us at the meeting. Jonah's neurologist saw us immediately upon arrival. When we walked into her office, she introduced us to her PhD student. Her student could barely make eye contact. That's when I knew something was wrong. Our neurologist picked up Jonah and examined him from head to toe. She was thorough. I started getting more and more nervous. Her PhD student didn't move. The neurologist finally sat down. Jeremy was in the corner bouncing the ball with Jonah, oblivious to what was happening. Mother's intuition told me that something very serious was going to happen. So the MRI showed results of abnormalities consistent with mucopolysaccharidosis. Took me a long time to say that. I couldn't get past her name. Our neurologist went to say what it meant. At the end of her deliverance, I sat there speechless, trying to wrap my brain around what she had just said. My husband, absorbed in playing with Jonah, had no idea what had just transpired. He saw the tears streaming down my face. Panic ran across his face. I asked our neurologist, "What are you saying? My son has a fatal disease?" She responded simply, "Yes." Reporters asked me, "How does that make you feel?" I'll tell you how that makes me feel, as if you can't picture it yourself. It's like a wormhole has opened up under your feet and sucked you and thrown you and your family across the galaxy. We're alone in the dark and helpless. So that's our initial response. And then we had to do several weeks of massive amount of testing. We did everything, echograms, his whole body x-rayed, a couple more MRIs, a sleep study, everything. We walked out of there with a fistful of prescriptions. And then we did more genetic testing to find out what kind of mucopolysaccharidosis Jonah had. It took about 6 weeks. So upon final diagnosis, which was Sanfilippo type C, I asked our geneticist again if this was a death sentence. She paused so long. I was so upset with myself that I had asked her such a question. And then she came back and she responded, "No. Today there are treatments for diseases that they never thought possible just 5 years ago." That's all she needed to say to me. My husband and I walked out of that office. We went home. I picked up the phone and I called my lawyer. And within 2 weeks, we had formed our foundation. Within 6 months, we got our 501c3 status. And then we started fundraising. We located and brought the handful of scientists studying Sanfilippo C to New York for a patient population meeting. With the help of our advisors, the scientists identified the best treatment options, and we went after them. Today, we are well on our way to seeing a treatment for Sanfilippo. We're at this point because our friends, family, and strangers supported us, and actors, handing over their hard-earned dollars to help us save our children's lives. Now I ask for your help. Please help us. Help the 1 in 10 Americans that suffer from a rare disease. Help us spur the development of treatments for rare diseases. The science is here now, and we can treat these rare diseases, saving countless lives. Like, who was it that mentioned the parents shouldn't be doing this? But we are. I funded all the science for this disease. Before I came along, there was no science for type C. We didn't even have a mouse model. And now we're this close to a treatment, and we've done it all on our own, nickel and diming it. So yesterday when I heard the proposal to introduce that Congressman Camp introduced to eliminate the orphan drug tax, it made me sick to my stomach. This is exactly opposite of what we're working for. Instead of spurring treatments for our children, we're kind of saying, you know, they don't need a treatment anyways, and it hurts. And so that's why we're here today, to implore Congress to help us find treatments.
Jill alluded to this, and so I don't need to say too much about how important it is for all families, for all rare disease conditions, to have supporters and advocates that are even outside of their own well-known family members, friends, neighbors, people who share their problems. And having a famous person as an advocate, all the better. And we are very honored to have with us today Johnny Lee Miller, who otherwise is known as Sherlock to me. I mean, we're on a first-name basis. And I promised Julia that because children are present, I am not going to say what is in everyone's mind, no Sherlock. I'm not going to say that. I'm not saying it. But Mr. Miller has spent tremendous time and effort in support of raising funds to treat Sanfilippo, including running a 50-mile, marathon, which is why his thighs are a lot thinner than mine. So Johnny Lee Miller, thank you and welcome.
I'd like to thank the representatives Lance and Crowley for co-chairing the caucus and hosting this briefing. And thank you to the RDLA and to Jonas Just Begun for inviting me and giving me the opportunity to speak here today. It's an incredible honor. Even though I am British, I cannot vote here. I am the parent of an American child. I still hope to be able to help and support legislative initiatives that spur the development of treatments for rare diseases. I am not personally affected by a rare disease. I became an advocate for rare disease patients when a camera operator that I work with, Jeremy Weissar, Jill's husband, asked me to support a local fundraiser to help develop treatment for his son, Jonah, as you heard. Without treatment, Jonah is unlikely to live long enough to see his 18th birthday. As a parent, I find it unacceptable that any parent would have to hear that their child has a fatal disease for which there is no treatment. Unfortunately, parents hear this far too often. Now here's a man standing next to me at work for 12 hours a day, carrying this terrible burden. I have a son the same age as Jonah, and I felt I had to help, but what can I do? I'm not a scientist, I'm not a doctor, I'm an actor, and not a particularly famous one, quite frankly. I'm working on that now. I realized I could raise awareness. I realized that this is terribly important. So, I run marathons regularly. I decided I'd use my next run to raise funds and, more importantly, awareness for Jonas Just Begun. Now, the support that we received from colleagues at work and the public was extraordinary. Once people hear about this issue, they cannot ignore it. So I helped raise nearly £200,000 by running actually 2 50-mile ultramarathons last year and turning over the contributions to the organisation. No, please. This money will help Jonah's Just Begun as they continue to fund research and hopefully lead to the development of treatment to save Jonah and other children like him. Quite frankly, I could run marathons in every spare moment I have. I will run more and I will keep running them to keep raising awareness. But this is only one piece of the puzzle. Too many children will have lost their lives in the meantime. I just hope, and I ask you to take a moment to consider what you can do to help patients with rare diseases. As a congressional staffer, think about the ways that you can assist patients with legislation to help spur the development of new treatments. There are patients here in this room that need your help right now. If we come together, doctors, scientists, advocates, parents, heroic parents, journalists, and legislators, we can solve these problems and we can find cures now. You have the power, much more power than us, to help save lives. Thank you.